thing that you have to do when you have a cell receiving a signal is you obviously have to receive the signal, so there has to be reception. Now we saw that in the hormone chapter, reception was very specific, right, Devin? Wait, why didn't you respond?
Me?
Yeah,
Respond to what?
I said, do you not understand that, Devin?
My name's not Devin.
Oh, okay, so let me get this straight. You heard me,
I heard you.
but he responded.
Yes.
So this reception is specific,
Yes.
just like hormones, right? Like, for instance, TSH, which stands for what?
Sorry.
Thyroid stimulating hormone. That hormone, thyroid stimulating hormone, ends up everywhere in the body. But who's responding to that? Obviously only the thyroid. That means that the shape of that hormone only matches something on the outside of which cell? The cells of the thyroid. So reception, which is a very specific process. Very specific. Right? You could say that if you look at like this little picture real quick, this could be called the target cell. And maybe this is the receiving protein, and perhaps this is the signal molecule that's going to end up going into that spot right there. So typically on the outside here you have a protein. This is your signal molecule. You could also say this protein is a receptor protein. Good? What's after reception? Transduction, which means you have to carry the signal inside the cell. Where are you typically going to carry it to? A lysosome? When you get a call from your mommy and you pick up that phone, how do you know that it's for you?
It says so on.
Isn't it your phone? And you know who that is, right? So you know what you're receiving. Where's the signal going as you hear it? Ear, connected to brain, and you're making like sense of this, right? If your mom said, "Don't drive the car anymore," and you still drive the car, like you obviously are making a decision based on that, right? So in our body, where is all the decision power? That's in the brain, correct?
Yes.
Where would it be for a cell? Nucleus. Now, what would we call it if you had like this next step? Instead of reception and there's transduction, then what's the last part? You receive it, exactly, response. You make sense of it. In transduction, you carry it to the center that's going to make sense of it. And then in response, you act on it. So, acting on a given signal. To act on a given signal. Right? It's pretty simple, right? Any questions? ABC. Okay. That's really about it for communication that I would like. I really want us to focus, lastly, just on maybe making sure we understand like that very specific concept. Brendan, how you feel about that? Why didn't you answer? Brendan's not my name. That's like saying that I have insulin, which can be picked up by about every body cell, but it's not really responded to by the cells of the spleen. Hopefully we all get that. The signal's everywhere. It's just you don't respond to it. It's like saying that there's a 96.5 radio station floating throughout this room. There's 106.1 floating throughout this room, and every other radio station that's in this area is floating throughout this room. If I had a radio, what would dictate which radio station is playing through that radio? Yeah, whichever channel I'm tuning into, essentially, correct? So that's kind of like the way these cells are. They have the ability to tune into unique channels when the signal's present. If I go into Louisiana, can I listen to 106.1? No, because 106.1 is a very local Dallas thing. I go into Louisiana, that's obviously not 106.1 territory anymore. The signal's not present, so I hence don't have a response from listening to my little shows. Good? Yeah, I listen to Kurt Craddock each morning. You don't listen to him?
No, I do.
You don't?
No, I do. Every morning.
Good. What's the next topic?
I have a question. For the functional groups, will we have to know anything besides, like...
What they look like, what their name is, and then, like, the fact that, like, dopamine would be an amine, or butone would be a ketone, or formaldehyde would be an aldehyde. Things like that. Sure. Absolutely. Right now? That'll be real quick. Julio? Sorry. Functional groups.
Please don't close this open.
Please don't quiz you over these tomorrow.
As you know, that's true.
So we'll quiz tomorrow over it.
Over what?
Or we can. It's really not that difficult to learn. Probably Griffith. What did Griffith do? What was his deal? Do what? Yeah. He's the mice guy. The mice guy. Wow, I just misspelled the word guy. Mice guy. Now, if you remember his setup, what he had was some living S bacteria cells. And this is case 1. He took his living S bacteria cells and he put it inside of a mouse. This right here is my mouse. It's this little box. He put his living S bacteria cells inside the mouse, and what happens is in the end you have a mouse that's dead. That's the result. So what does that mean about living S bacteria? Was it good or bad? Obviously, this living S is going to be bad. Cool. So then in case 2, what he did is he took living R bacteria, which he knew himself was like actually an okay bacteria to mess around with, and he took that living S bacteria, which was okay, but you put it inside the mouse, and what happens to the mouse at the end? The mouse is alive. Perfect. Now, in case 3, or in his third attempt, what he did in his third attempt is he took some S cells. So he took the S bacteria, which typically causes what? Death, but then he killed it. That was heat shocked, which is a nice way of saying he killed the bacteria. So what happens after he heat shocks the bad bacteria? He'd stick it inside the mouse, and what's the outcome? And the mouse is alive still. So that means that this agent that was in the S bacteria that causes the issue is only an agent when it's inside of a cell that's living. And the question was at this time, what's the agent? Now we know today it's DNA. He didn't know that necessarily. Make sense? But then what he did is he took kind of the last 2 cases into account real quick. He took some living R. And does living R kill you? No. But he added to that, so plus some heat shock S. Now does heat shock S kill you? No. But when you mix them together, you stick it in the mouse, you have a dead mouse. Just like there's that dead cat back there right now. True story.
Is he open? Is he closed?
Open.
Wow.
And he's okay. But just like that cat's dead, this mouse would be dead.
Why is that?
Because the agent that was from your heat shocked S, which we now of course know is the DNA, was incorporated by transformation or transduction. I guess it would be in this case transformation, not transduction. By transformation, the heat shock S was dead and then released its DNA that was transformed into the R cells, taken out by the R cells, which were living. And now the R cells have gone bad on you. Do you follow this logic? Now, it was at that time that he knew that whatever this agent was, according to Griffith identified that there was an agent of inheritance, and he identified the agent as being protein or DNA. Now, why did he say protein or DNA? One is obviously he didn't know, correct? And remember that a chromosome itself is made of what? DNA plus associated protein. So like, you wouldn't know which it is. The next people to talk about. Hershey and Chase. What did they do? What is their story?
Holding stone for their DNA.
Yeah, they were very definitive in DNA. They used what? They used radioisotopes, both of type sulfur and phosphorus. And they established from this that DNA was the only genetic material. Remember that the P is in the DNA, but the S is only in the protein. So they were able to basically redo something like Griffith's experiment, but trace and find out that whatever was causing the transformation contained only phosphorus, which means it had to be the DNA of the nucleic acids. Cool? I also like taking this time to talk about Chargaff's rules, which are so simple. Chargaff and his little rules, which he said A amount has to be equal to T, and the G amount has to be equal to C. It's like so simple to get. Very, very, very nice little questions.
Wait, and then U replaces the T, right?
Yes, in RNA. There is no T in RNA, there's just U. Rosalind and Franklin was all about X-ray crystallography, which showed like maybe a possible like structure on the inside of the DNA ring. But it wasn't until you had Watson and Crick that used some of Rosalind Franklin's ideas, and they concluded that you had DNA that was double helix, double-stranded, running anti-parallel. And not only did they come up with the structure, but what else did they come up with? Right, a mechanism of reproduction, right? A mechanism that could get you multiple copies. And what type of model is that? Is that a conservative, dispersive, or semi-conservative model? Semi-conservative, which means that out of your original DNA, remember your DNA looks something like this, right? And in the end, this one piece is going to end up being two pieces. How many pieces of DNA do you see there? Two, right? Used to just be one between these two that I'm pointing to right here. There used to be just one DNA running in between those guys right there. But now there's two because each acts as a complement. That's what semi-conservative means.